The overall goal of the following procedure is to assess brain remodeling in hyperglycemic conditions and to follow radiolabeled molecules in zebrafish.
Hyperglycemia is characterized by an excessive concentration of blood glucose. Chronic hyperglycemia may be due to diabetic conditions and causes different vascular dysfunctions that may in turn trigger clinical complications such as myocardial infarction, stroke, or diabetic foot ulcers. Also, acute hyperglycemia due to stress may promote hemorrhagic complications in ischemic stroke.
Zebrafish is an interesting model for understanding human diseases and their impact on the central nervous system. It is particularly true given that the brain of adult fish displays an intense neurogenic capacity and an outstanding capability for brain repair mechanisms compared to mammals. Therefore, zebrafish represents a good model for investigating brain remodeling following a stress under acute and hyperglycemic conditions. It also allows investigating the biodistribution of radiolabeled molecules and potential therapeutic agents.
All experiments were conducted in accordance with the French and European Community guidelines for the use of animals in research and approved by the local ethics committee for animal experimentation. Catch a fish gently with a fish net. Put the fish in a small volume of tricaine anesthetic, diluted at a final concentration of 0.02% in the water. Wait until the fish stops moving. Remove the fish using a cut pipette and place